Gene.iobio is a web-based application from the iobio project, developed to provide simple visualization and analysis of genetic variants, identification of potentially interesting variants, and an understanding of the underlying data. In this quick demonstration, we will explore the basic interface and functionality. To begin, we select a gene. This can be a protein coding gene or one of many other genetic features, for example pseudogenes or link RNAs. Once selected, the canonical transcript is displayed, with the coding DNA sequences, start and stop codons, and UTRs depicted. Next, select the variant and alignment data for a single sample. For this demonstration, we'll be using the example Platinum Trio dataset. Pressing Load begins the process of real-time analysis of the data prior to being loaded into the visualization cards that now become visible. All of the variants from the selected file are displayed, colored according to functional impact and with a shape corresponding to the variant type. Hovering over an individual variant displays detailed information about it, including the sequencing depth, which is visualized in the track below the variants. Clicking on the variant locks the information in place, enabling the user to link out to additional resources. This can then be released by clicking Unlock. The Filter Variants card allows custom filtering to be applied to the variants. Hovering over any one of the labels highlights the variants with that property. By default, the variants are colored by the SNP-F impact, but they can also be colored based on the annotated effect. Clicking on Labels locks the filters in place, allowing for multiple filters to be applied at once. For example, only UTR insertions that are not observed in the 1000 Genomes project can be quickly displayed. The Rank Variants card ranks the variants in order of significance, with the leftmost variants in the table more likely to be functionally interesting. Each row represents a single piece of information, for example, listed as pathogenic in ClinVar, and the higher the row, the more important it is deemed to be. The order of the rows can be modified to reflect the user's views on importance. For example, the 1000 Genomes allele frequency row can be moved up the table, increasing its significance. Having performed this change, the most significant variant is still the ClinVar variant, but now, being a private mutation, is more significant than being annotated as of moderate impact. Individual variants in the table can be selected to highlight them in the other cards. Now we'll change to the DSP gene and see how quickly the data is reprocessed and visualized. We can now click the Call from Alignments button to call and annotate variants directly from the sequence alignments in real time and display only those variants not already seen. In this case, 2 variants not present in the original file have been discovered. To fully explore the power of gene.iobio, investigate trio data, and explore some use cases, please watch the accompanying screencasts.